This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 83, recorded on February 12th, 2015. Hello, everyone. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello there, Vincent.
And Daniel Griffin.
It's good to be here.
And, you know, now I have three.
It's three.
This is called TWiP Reboot, by the way.
Oh,
Do you know, Daniel, since you were the pop culture person and not Dickson, do you like Star Trek?
I do, actually.
So they did a Star Trek reboot with J.J. Abrams, the director. The last two movies, they call it the reboot because he actually started at the beginning.
Okay.
And he infused it with a new sense of purpose, and it's really good. So we're going to call this TWiP Reboot.
Why not?
Because we have a new person who has infused it with a new sense of purpose.
Exactly right.
And I can just take a back burner.
We're going to kick it up a notch.
Really? Where are we going?
We're going to elm roller gas this show.
So actually, I had plans today, Vincent, to sort of pull you more into the case discussion. So we'll see how that goes.
Excellent.
I can do that. I like cases. I did. So last week I taught first-year medical students microbiology and infection course. You must know that course here. Mm-hmm. It's taught by Rachel Gordon.
Okay.
So usually I give 5 lectures, and this year she said, let's have you record 4 of them, and we'll do a session where we ask them questions. We basically made up cases.
Nice.
Oh, that's great.
Nice.
And then I would give them questions.
These are first or second year students?
These are first year students. And I had 4 of the students in the class had been in my virology course.
Ringers.
At Columbia, and I saw them out there, and I pointed, all right, you and you and you. And so, for example, they were based on my own experience. So one of them had to do with the ER doc here at Columbia who came back from Western Africa.
This would be Craig.
And contracted Ebola, right? Remember that? So his friends were quarantined. He had gone bowling the night before, and his friends were quarantined. And there was a movement to have them released by lawyers, American Civil Liberties Union.
Yep.
Okay, an organization I like to support.
And I got involved
Sometimes.
To a certain extent. So I told them that story, and then I said my advice would be no quarantine. And then I asked him some questions. You know, why not? Oh. Yeah, I asked him a question saying, why shouldn't we quarantine them? And gave them a bunch of possibilities. And the one, of course, is that no one has ever transmitted Ebola infection without being really sick.
Right.
And the other one was, because we have antivirals and we can treat everyone. And 10% of this class picked that one.
Really?
Oh. They think that we have antivirals. That we can.
So had any of those attended your class?
No.
Right.
Okay, fortunately not.
Like last year, I didn't talk about Ebola. Anyway, the point is that I like the clinical. I think you engage in a different way when you ask clinical case-related questions. And so for the medical students, it's quite important because they really get into it.
Yeah.
Right.
Yeah, we've noticed that when you're trying to teach a topic to people who will one day become doctors, or even people who are already doctors but going for further training, if you start with a clinical vignette, if you give them a person, it suddenly raises the interest level because everyone is hoping that they're going to be able to step in, figure out the mystery, help this person. And so it goes from just, is this going to be on the test, to I need to pay attention. Because this is a person, this is a human being, and it gives focus, I think, to the learning. Exactly. And hopefully our cases are a little reward for people that are listening to TWiP.
Yeah.
Well, we didn't get any responses to last week's case, which I guess is because we're only recording a week afterwards, right?
Yeah, even less than a week. There's a delay, and
so I post on Saturday, and so we don't have. So what should we do? Save it until we get some responses?
No, I think it would be good. We can go ahead and discuss, and I'm hopefully our readers will listen. They'll be thinking about it. We may not have gotten some emails, but Dickson, I'm sure, will have some ideas. Vincent, maybe you all have some ideas.
I have no ideas about anything.
You have great ideas, Vincent.
Oh, why are you trying to suck up to me?
No. Because you think I'm in your doghouse, don't you?
No, I don't at all. Just because you did a bad thing yesterday? Not at all.
I didn't do a bad thing yesterday.
No, I think...
Guilty with an explanation.
No, I think this last case is very gettable. I was presenting this case to my dad over breakfast this morning.
He got it?
He got it.
Excellent.
So,
is he a doc?
No. Real estate developer.
Right.
And so I think this is very doable.
You have to remind...
And I'm wanting to present cases initially that, here are the clues and you can get it. And we're going to do that again today.
Right.
But then we're going to move into these really challenging
fuzzy zones
where you may not get it, and then we'll return to some common ground.
So Dr. House type cases.
Yes.
Oh, House. You're a fan of House, aren't you?
I was, but...
Daniel, what kind of bow tie do you have today?
Now, today I have my influenza bow tie. And as one of my lab members commented, there should be 8 little RNA segments. There's only 4. So the artist needs to work on that. But no, and it'll be when we start discussing our paper. The reason why I have gone with influenza will become apparent.
Interesting.
Daniel, of course, has his new microphone today. So
we're both.
This is the first time for you on TWiP.
This is high tech.
You did a TWiP with your mic last week, but you should both now sound exactly like me.
Yes.
Right? Mellow.
That's right. So we will solve last week's case, and if people send in some solutions, we can read them next time quickly.
Good.
Right?
Okay.
And I think we just have to make sure that if we're going to ever give an award or a prize or something like that, that your email has to have been sent to us prior to the posting
date.
of
That's right.
The solution.
Yes. Yeah, because otherwise
it's really easy.
Well, I'm not going to post this episode till Saturday because it's only been a few days since the other one went up, and people don't need too much TWiP.
No. Yes. You can even delay it until next week, and then they won't even know that we recorded it.
Remember, it was called This Week in Parasitism, but we have rarely been week to week. We have rarely been week to week.
Well,
shall we start? Shall we go ahead?
Yeah. Let's do this.
Let's now, Dickson. You remember? Do you want to recap? Do you want to recap? Who was this person? How old were they?
No. I want you to read the case history again.
I realized I didn't bring my notes, so this time I'm gonna wing it. But it's okay. As our listeners may remember, this is an 18-year-old young man.
Right.
Who was having these episodes. He had 2 of them where he felt just fine.
Oh, yeah.
And then started to feel not so good. Had the onset of shaking chills. Actually described his teeth.
Right.
Actually chattering, right?
Yeah.
And then developed a high fever.
Right.
Didn't want anyone to touch him, but we pointed out that's because it was his mom that was trying to come in and take care of him. If it was an attractive young lady, maybe he would have had a different response.
Possible.
Went to bed,
and
this was a very short-lived fever, and then he felt a little bit not so great, but by the next morning, completely fine.
How about that?
And then initial fever, I think, was Monday. Tuesday, he felt fine. Wednesday, same episode. Thursday, mom says, you got to see the doctor. Friday, he goes to see the doctor.
Right.
And we elicited a travel history. He was down in Central America.
Yep.
And he may have been bitten by...
a vector type of thing.
By a vector.
He may have had sex, but he said no.
He may have had sex, which we discussed is often increased when people travel.
Right.
And...
so there we have it.
Yes.
Basically it.
Okay.
And we mentioned that he took his malaria medicine.
He did
and
do that,
did exactly what he's supposed to. Yeah. So he tells us.
And remember how long ago it was that he went?
It was in August, and he is presenting to the clinician in February. So,
so he's well beyond taking his anti-malarials at this point.
So, Dickson, what are you thinking? What diseases come to mind?
The last time we talked, which was when you presented this, of course, I couldn't think of anything else except malaria that's consistent with this diagnosis because of the pattern of fever, first of all, and chills. So that's called paroxysms.
Okay.
We know what they're called. They were described by Hippocrates quite a while ago, actually. So we know them. And we know that there are quartan and tertian types of paroxysms. So this, and we also know that there are species of malaria that can linger in the body for a long time, silent, sort of hypnozoite-like, hypnozoitic-like, I meant to say. And it's due to the fact that they produce a stage called the hypnozoite. This is in Plasmodium vivax and Plasmodium ovale, which lives in the liver, and it's non-dividing at that point. It's sleeping. It's cryptic. And then all of a sudden, sometime later on down the road, one of these decides to start reproducing, and the next thing you know, you've got a chills and fever, and you've got malaria, and you wonder, well, how could I possibly have malaria? Because I took my antimalarials. And the answer is because antimalarials don't prevent infection, they prevent disease. All right? So in this case, though, my logical mind, since I taught this darn thing for 30 years, says if it's not malaria, then it's a pretty weird parasite.
Okay.
I think it's AIDS.
You're going with AIDS?
Yeah, he had the initial clinical syndrome back then up to a year, and now he's slipping into low CD4 counts.
Okay.
He's got some opportunistic infection.
Well, how do you explain the next day he was feeling better then?
Next day of what? It's this latest
No, he
episode?
had chills and fever. He went to bed miserable. He woke up the next day, he felt great.
I don't know. But since it's so
I'm
long,
actually glad that Vincent brings up HIV. He's wrong, but I'm glad that he brings it up.
because eventually we'll get to that.
No, because it's one of those things. You read in the newspaper. I think we read just yesterday. Oh, isn't it great that Ebola's over? And now everyone to the CDC website where you see rising rates in Sierra Leone. But now that it's all over, and they often say that about the HIV epidemic. But HIV, there are over 1 million people in our country living with HIV.
Yeah, we were talking about that this morning.
50,000 new diagnoses of HIV every year in the United States.
That's remarkable.
And globally?
Globally, we're over 30 million.
And how many new infections each year?
Oh, I'm not sure the exact number globally. The deaths are over 1 million. So
Total deaths
from HIV are over 1 million per year.
But not from AIDS itself, from an infection that AIDS allows to happen.
And that confuses numbers. A large of those numbers is you have the HIV infection, and then you come down with tuberculosis.
So hang on a second. Maybe Vincent and I are both right.
So I like Vincent bringing that. I think that's important to bring up.
He could be right, and I could be right too. I mean, this could be an HIV patient with malaria.
I don't have to be right.
No, and that's
actually. Wait a minute. He's saying this on the air.
Do you get the incredible that I always want to be right?
This is incredible. Well, you like being right. Let's put it that way.
I work hard to get things
Who doesn't
right, but I don't
like
have
to be right? But
Daniel, can you tilt your mic down a little bit?
Yes.
And if you're going to...
I'm starting to slump, so I need to
if
adjust my
I don't
mic
know what
every
your
time I slump.
final position is going to be. Also, when you turn to Dickson, you go off mic. Because these are very directional.
We're having a little...
I will stop turning to Dickson.
So in the future,
him
if you want to get eye contact, we can put him here in the future.
That'll work.
Okay. No, this might be better. I might actually prefer
He wants to avoid that.
You want to avoid eye contact? You do look at him a lot.
I resemble that remark.
See, I can look at you both without going off mic.
But notice that I can sidestep my mic and still be in mic and still look at him.
That's because he's doing...
I know.
You're doing. That's okay. He's gotten used to it already. But these side things are not picking up anything.
Right.
This is an end-on mic.
You're going to ask us now, what additional tests would we like to see?
Didn't we get a CBC, or was that unremarkable?
We did get a CBC.
He had bilirubin, elevated bilirubin, as I recall, in his urine.
The platelets were a little bit down.
And he was anemic.
He was a little anemic.
So this has a strong possibility of being malaria.
Okay.
No, and that's actually so. Before I tell you what it is, I'm going to go ahead and say that in a returning traveler, you always want to focus on what might kill them. And so a returning traveler, even, oh my gosh, we're seeing here how many months out? A number of months out.
Right.
This returning traveler is having a fever. He's having symptoms that could be consistent with malaria. Really, the first thing you need to do to be safe is to address, could it be malaria?
You know what? I think we did have one listener answer.
Oh.
I'm sorry to interrupt. Would you like to hear it?
Yes.
Oh, that would be great.
Because Robin is our retired doc who always answers, and he answered right away, and I thought he was talking about the previous week. So he says malaria, shaking, chills, and fever followed by sweats, not specifically mentioned in this case.
Right.
So that sounds like it is a characteristic of malaria that is unforgettable once one has had it. I had malaria 4 times.
Wow.
Thick blood smears is de rigueur. Some helminths will also produce a similar syndrome. Forgot which ones.
Well, we could refresh our memory.
And then
on
he
that one.
gives a list of things every physician should have been taught to familiarity, not necessarily competency or proficiency, because you're always talking about getting in. And these include the use of a microscope, Wright's stain, blood,
Here.
thick and thin, Zeal-Nielsen stain, sputum, Gram stains, all and sundry, Scotch tape for ova, hanging drop for trypanosomes, potassium hydroxide prep for hyphae, a Wintrobe hemocytometer manual CBC, spun hematocrit, silver stallion sigmoidoscopy, endotracheal intubation, indirect laryngoscopy, reduction of easier dislocations, and slit lamp examination of the eye. So he
Right.
thinks every doc should know how to do these
Wow.
things. So
Okay.
Daniel, today new docs learn how to do this stuff?
Now, when I trained, we did. I was listening to this list, and but I think, unfortunately, these skills are being lost.
Yeah.
And I say unfortunately, but if you're practicing in the United States in, we say, a high resource area, most of those techniques you won't necessarily be doing anymore. The sigmoidoscopy he mentions, when I started in private practice, my day would start. I did one every morning, and that was the way we kept the screening. But now, as we screen for colon cancer with full colonoscopies, that's moving to a certain group of people. Again, not something everyone would do.
What's silver
the
stallion?
KOH
What does silver stallion mean?
That I don't know.
All right.
We'll have to Google that.
Well, maybe we can ask him to respond.
Yes. No,
Start
we
a
may actually get a
conversation.
Google response in time. I mean, certain other things I think are ashamed of the fact that
the rigid scope is lovingly referred to as the silver stallion.
that predates me.
Because that was a rigid scope that you could only put in so far,
Yes.
right?
Right.
Yes.
Right. Ouch.
Silver stallion.
Right.
Yeah.
Yes,
I never had any experience with the silver stallion, so I'm
So now you use a flexible tube with optics in it, and you go all the way through the
a
colon.
little joystick, so you're able to turn left, right,
yeah?
up, down, and
Nice.
you advance. And you can get them where you actually look through the scope, but it's a lot nicer, I think, and you're able to keep your face a little bit farther away from the instrument by projecting it onto a screen.
Yeah.
So
Right.
I would do that.
My first colonoscopy years ago, they kept me awake, and it was up there, and the guy said, "I'm going to give you a quiz when we're done, so pay attention."
Duh. I was awake too, but I took the La La Land drugs so that I knew I was there, but I didn't actually know I was there. I was sort of like out-of-body experience looking at myself getting a colonoscopy, so it was very interesting, I thought.
All right. So he thinks it's malaria.
Okay.
So now you're going to show us a lab.
Now I'm going to show you,
test.
and I'm going to explain. This is going to be I'm going to fold this over so that
I'm going to put my glasses
so that
on for
Dickson
this.
can see a representative smear, and I'm going to explain why it's representative. I bet Dickson has a good idea why it's representative. So I push this over
Just
to
the
Dickson
master
so he can
of
look
the
at
smears,
it.
aren't you, Dickson?
Oh, yes. You see,
And I'm going to describe. Should I describe what Dickson is seeing, or does Dickson want to describe it?
I could actually say what I see.
Okay. Let us
So first of all, I see a whole bunch of red blood cells. They're not quite all touching each other, so we're in the feathered end of a thin smear, not a thick smear. This is a thin smeared blood sample. It looks like it's stained with Giemsa.
So what's the difference between a thin and a thick smear?
Oh, that's a very easy one to do. So a thick smear is actually not a smear. It's a drop of blood that's allowed to coagulate and dry.
Right on the slide?
Right on the slide, and then it's dipped into an aqueous solution, which lyses all the red cells after they're fixed. So it actually lets you look through a whole bunch of layers of red cells to see the one or two parasites that might be there. It's a way of detecting very low levels of parasitemia. This, on the other hand, is not a low level of parasitemia.
That's a thin smear.
This is a thin smear, and I see 1, 2, 3, 4 things, which of course I knew what they are. I see 2 things that would qualify as a signet ring stage, which is the earliest stage for malaria. And then I see some later signet rings, which would be called early trophozoite stage. And not only that, the red cells that are infected with the late rings or early trophs are much larger than the average red cell's diameter. And this is a tip-off. And then there's another tip-off here, and it's pepid. Looks like it's got measles. The red cell looks like it has measles, in addition to the parasite.
Yeah, see?
Yeah, sure.
It has dots.
Little dots
that have a
Little red
special
dots.
name.
Oh, look
They're
at that.
called Schuffner's dots.
They do
with
have
an
dots.
umlauted U.
All these things you said, all I see are red blood cells. I see two of them that look different.
Those are the different ones. Those are not.
And then there's a couple others with weird nuclei. So the signet ring is that like that C structure?
It's like a little
I'm
ring
going to
with a
lean
dot.
forward so we can point. Yeah, if you set it down here, we can
I see
actually,
the signet is like that
That's
one.
it. And there's another one down at the bottom.
Is
the
that
ring
there?
form in there.
That's
There's
right.
a ring form in there.
So
Got
you've got
it.
2 rings and 2 late rings or early trophs.
Got it.
And because the red cells are enlarged, and because they have Schuffner's dots, which actually are indentations of the red cell membrane, so they collect the eosin stain from the Giemsa, this is a pathognomonic diagnosis
For
right
what?
here.
For what kind of malaria?
Plasmodium vivax.
Vivax. Now,
Classic.
But Dickson, he took his malaria
Classic.
medicine.
I told you.
Tell me again, because
I will tell you once more, and I will repeat this, because what was the medication that he took? Let's say he took mefloquine, okay? Mefloquine. What does mefloquine attack? It attacks the blood stage only. All right? Now, if he had taken another drug with it, which the British often do, called primaquine, that would have attacked the liver stages. However, there's a problem here too, because primaquine only attacks the dividing stages of the parasite, even if it's in the liver. So it's possible that even with primaquine, you could avoid being malaria-free, and at the same time, these hypnozoites, which are non-dividing stages, like trying to treat a latent viral infection with an antiviral, because the virus is not replicating, therefore the antiviral can't work. And the same is true for these parasites. When they're in a dormant stage, there's nothing going on there.
But why are these drugs even used then?
Because what they do is that they ablate they blunt the infection and keep it from going on to developing into a disease. Okay, so if this had been Plasmodium falciparum, which has no latent liver stages, that drug would have probably wiped out the infection.
So
I think that's a great question that you ask, Vincent. And a lot of people ask, well, why do we take malaria prophylaxis if this can occur? If Dickson is telling us
if indeed he's speaking the truth.
And he is. Dickson is speaking. He often speaks the truth. And in this case, again, he's speaking the truth. We're really giving malaria prophylaxis for Malaria falciparum.
Correct.
I pronounced it falciparum until I met Dickson, but
I used to say falciparum until he tried to correct me. And I still say falciparum.
So I'm not sure not sure who wins on pronunciation, but with malaria, we're concerned about people dying from malaria.
Right.
And falciparum is responsible for most fatal cases of malaria. And I say most because some deaths due to malaria can be due to Vivax, can be due to Ovale, can be due to malaria. Or what's the new?
Nolzi. You
I
like Nolzi?
Nolesi, but you guys call it.
Well, there was a wonderful article that we cited once about deforestation in Malaysia and the encroachment into those zones that used to have just primate malaria, which was Nolzi.
Yeah, that's
and
right.
now it jumped into people.
Yes. So let's focus a little bit on diagnosis before we get back into this really, I think, important life cycle. So the diagnosis, and I made the mention that I'm showing a representative blood smear because, as Dickson points out, you want to start off with your most sensitive test. It takes quite a high level of parasitemia to be able to easily pick up multiple intra-erythrocytic forms on your thin smear. So we start with this thick smear where you really have a stack of lysed red blood cells, which actually takes a while to dry. So you're waiting for this thing to dry. But because you have so many red blood cells, you can actually pick up that there's some form of malaria in there. But you can't actually see the dots. You can't see the characteristic morphology of the forms. So the actual telling, what species it is, often comes from looking at the thin smear.
Right.
And when you look at someone who has Vivax, people with Vivax are going to have very low parasitemia levels. We're talking less than 2%.
Right.
The only forms of malaria that are going to get you up to 10% or more are going to be your falciparum, falciparum. I don't know how we'll pronounce that from now on.
Falciparum.
And your Knowlesi.
Knowlesi.
And those can get you 10. They can actually even get as high as there's been cases of 50%.
Yeah.
Those you're going to see right away on a thin smear.
Sure.
But for something like Ovale, Malaria, and Vivax, the thick smear is going to let you know something is going on. And then there's these forms, which we talked about, the fact that you might have these dots, or you might have deformed red cells, or you might actually have a ring form that looks like a set of headphones, like the headphones that Vincent's wearing, which would be falciparum.
Or probably falciparum.
There are some falciparum
cases that I've seen that also were Vivax with double infections.
So let me understand. He acquired this when he was, what was it, Guatemala?
Yes.
He was in Guatemala back in August.
And he didn't feel badly until the present time.
True. And we're going to talk about why in 33 seconds.
Okay.
Right.
The last thing we're going to say, so here you are, you're at Columbia, and you have a case like this. You're worried about malaria. The guy's sick. It's 2 a.m, and you're general medicine. You're not sure, so you want somebody to look at that malaria smear. So you call up the pathology fellow or resident and say, I'm really worried about malaria. Can you come in and look at the smear? And it's 2 a.m, and so he rushes right in. Of course, actually not. What does he do? This is the modern world, and the modern world, and this isn't just here in the U.S. We're now doing in the U.S. what they started doing overseas. They pull out this little test, and it's almost like a pregnancy card, and it's called the Binax Now. And what we realize is that if you have malaria in your blood, you have antigens of that malaria, and you can actually use, it's a sort of a pocket little drop of blood ELISA test to see if there's any malaria in the blood. And it is very sensitive and very quick.
Right.
And I actually brought a little picture for everybody, but I will describe it. Most people are familiar with the pregnancy test, where you dip it, each urine in that case, usually the ones that people are using at home, and the urine moves up, and when it crosses these areas where there are antibodies impregnated on the test, if the right antigen is there, a line will come across.
Right.
And it's just, this is the same. There's a line for whether or not it's falciparum, whether or not it's Vivax or Ovale, and then there's a control strip.
Nice.
So in this case, if it runs through and just the control strip comes up, we'd say we haven't detected malaria. If both come up, it could be a mixed infection. If just one or the other comes up, then you can tell. And that's actually what they'll do here at Columbia if you call in the middle of the night. And it's a great advance to be able to very quickly make a diagnosis. But again, it has only a certain sensitivity.
Yeah.
So you need to sort of realize, even though it's negative, you can't say, oh, it's not it.
So that was done in this case.
And that was done in this case. And it came up with the single band, and it was Vivax.
Vivax. All right.
So when I was
now we get to go to that great case. Is how did he was there, he did what he was supposed to. The doctor told him, take your malaria meds. How did this happen? And this gets into this wonderful life cycle that we get to talk about. The fact that your malaria medicines prevent you from getting sick with malaria, but they don't actually prevent the infection. I mean, the way to prevent the infection is bed nets and long pants and staying inside at night and DEET and things like that. But with malaria, the mosquito bites you. We talked about it's the 1 in 100 mosquitoes that likes to bite twice. It's the Anopheles mosquito, and if you're being bit, you can look at it. And I know Dickson and I have talked. You could actually tell. You don't need to know much. If it's nice and parallel to the skin,
Vincent knows
it's
this.
okay. But if it's at a 45-degree angle, right?
Yeah.
You'll never sleep alone anymore. You'll be looking at the wall. Is that
That's what you said, right?
You may want to just swat all mosquitoes instead of doing this inspection. But after the show, you may want to pause and look at the angle, look for the speckles on the legs of your mosquito. Right?
And on the wings.
And on the wings.
That's right.
And the mosquito goes ahead and it bites you, and it injects something into your blood, right?
It does.
And what are those?
Well, they're haploid sporozoids.
Yes. And now these then go initially to the liver. And in the liver, they can do 2 different things.
Yep.
And it depends on the strain of malaria.
Yep.
If it's Ovale or Vivax, it can do 2 things. If it's the other strains of malaria, it's stuck with just doing one thing, which is a replication inside the liver cell. Then it goes very shortly, within a few hours after it leaves the liver, gets into the red blood cells and starts its cycle.
Right.
Now, a lot of the malaria drugs don't even work until after it's been through the liver, until after it's in this erythrocytic replication period. So when people come back, I've been on my trip and I feel fine. I don't think I need to keep taking my malaria medicine. Well,
Right.
they may very well have malaria replicating actively in their blood. So a lot of the medicines you need to take, like mefloquine, like doxycycline, you need to take for 4 weeks after you get back to make sure you completely kill all those
Did he do that?
stages in the red cells. Now, this person actually did, but we're going to discuss why that didn't save him. Because with Vivax and Ovali, when it gets delivered, it has a second choice. It doesn't have to replicate. It can go into this dormant hypnozoite stage.
Exactly.
And I think that's what happened in this gentleman. And there, as Dickson mentions, there's really only one drug, it's kind of scary, that treats the hypnozoite stage. So if somebody's been in an area for a prolonged period of time, or if you need to make 100% sure that they're not going to get malaria and they've been in an area with Vivax or Ovali, you need to give a medicine that is going to treat the hypnozoite stage. And that medicine, as Dickson mentioned, is Primaquine.
Yeah, so there's a caveat here too, though. In fact, the episode that championed this was on MASH, one of the episodes of MASH, because they discovered that a lot of Italians or people living around the Mediterranean basin have a condition called glucose 6-phosphate dehydrogenase deficiency. And if you give Primaquine to those patients, you will exacerbate a hemolytic event, which is not good, because that's what the malaria parasites do also. So they found that out by giving Primaquine to people with glucose 6-phosphate dehydrogenase deficiency. And they almost died from that rather than from the malaria itself. So who's the Italian on MASH? Why am I looking at Vincent when I ask that question? No, he was, what the heck was his name? He was a short guy. He was really nice, fun to be around. Radar. His name was Radar, I think.
No, it was
that Radar?
Well, there was the guy who liked to dress as a woman
That's the one.
to get out of the
and
military.
Of course, that never worked.
Yeah.
Because everybody was dressing as women in the military in those days.
Yeah, Radar was the assistant, right?
Yeah,
Radar.
it wasn't Radar.
Radio operator?
Not Radar. It was the other guy. And
Oh, I didn't know. You know, I'm looking this up. And of course, I come across Alan Alda,
yes,
whose real name was Alfonso Joseph d'Abruzzo.
really.
And he had polio when he was 7.
Oh, wow.
What a remarkable coincidence.
He happens to be 79 at the moment.
So,
Wow. That's why he's interested in science communication, I guess.
so. So who was that Italian?
I didn't find it.
They don't have a complete list of characters.
What is it? MASH, M-A-S-H.
M-A-S-H, right. It's a mobile
I never
army
really- I
surgical
never really-
hospital.
Got into it. Here we
Yes.
go. MASH TV series. Who are the characters?
Cast of characters.
You want the actual names, not the actors, right?
No,
recurring characters.
Yeah.
Okay, here we go.
Yeah.
What do you want me to say? I got a whole list of them here. Do you want
How
to have?
many you got?
Well, I'll go through them.
Okay.
There's Hawkeye Pierce, right?
Hawkeye.
Hot Lips.
Hot
Houlihan.
Lips, of course.
Maxwell Klinger.
No,
John Patrick Francis Mulcahy.
not him either.
Trapper John.
No.
Henry Braymore Blake. Franklin Marion. Frank Burns. Walter Eugene Radar O'Reilly. E.G.
Yeah.
Honeycutt, Sherman Tecumseh Potter, and Charles Emerson Winchester III. That's it. Those
That's
are the
all they
main.
list?
There's no Italians. Listeners, tell us.
This
who
is
the
wrong.
Italian-
Yeah, we may need a listener. It wasn't Klinger? Klinger wasn't the one?
Klinger.
It was played by an Italian. It was an Italian character.
We don't know, do
Okay.
So Dickson is right again about the G6PD deficiency, and
I had
and
coaching.
that is a concern. So prior to putting someone on Primaquine, you actually have to look for this G6PD deficiency.
Interesting.
And if they do have it, again, as I mentioned, well, we only have one drug, and
What do you do?
now I'm giving medical advice, I guess. You can probably use a low dose of Primaquine.
I see.
Because the dose that we use was based upon, I think it was actually a military study where it was post-World War II. A lot of people actually acquired malaria during World War II. A lot of the men who were touring the world on, what they say, Uncle Sam's nickel, right? And that's when they came up with give this what probably is a rather large dose. So you can perhaps get away with a lower dose, careful monitoring to avoid the hemolytic anemia that'll induce.
And what
The claim
happened with
is
our
that
patient?
more people died of malaria in the military on both sides
Yes.
than died from bullets.
No, that's true. And in this case, we have two challenges. One, we have acute malaria that we need to treat.
Right.
And two, we have evidence that there's a hypnozoite stage, so we need to treat that.
But if you didn't treat him, what would happen?
The same thing again sometime down the road.
Over and over.
Total 5 years. It's good for about 5 years, and then after that, it burns out.
Yeah. And that's actually true. I think there's this myth of people 30 years later saying, "Oh, I'm having my malaria again. You know, I need to go to bed." And no, I don't.
Let me have my gin and tonic here.
there probably is a limit in most cases to how long you can milk the recurrences.
So what happened with our patient?
So our patient was treated initially for the malaria, and then actually went on and took a course of primaquine to wipe out the hypnozoite stage, and he did great.
How would you know?
We followed him up. We checked and asked him how he
And
did.
at least blood forms were gone.
Send him Christmas cards.
And he felt fine, right?
He felt fine.
Yeah.
Okay.
Yep.
Wonderful.
By the way, it's worth pointing out that although malaria infects millions and millions, perhaps even 1 billion people, it has a less than 1% mortality rate. But because of 1 billion people, that's a lot of people.
Yeah, they say 300 million cases of acute malaria every year.
Those
but
are the
then
cases,
the
that's
number
right.
of deaths is below 1 million.
It's very low.
And
But it's
so-
still very significant, of course, but it's still...
Well, it affects your life, right?
Oh, yeah. And
Makes you non-productive.
the other thing is that I know these studies have probably been brought up to date more recently, but recurrent malaria. There's a variety of Plasmodium falciparum malaria called cerebral malaria, where you actually go into a coma. And then if you're fortunate enough, of course, you wake up. Sometimes that's what is the cause of death. But as you wake up and then go about your daily life and you acquire it again and you go into a coma and then wake up and then go about your daily... If you get 5 or 6 of those in a row as a small kid, your ability to cognitively work your way through problems is diminished greatly. So there is a price to pay for this.
So our one respondent, Robin, had malaria 4 times.
4
Now,
different
why
times.
is this? Don't you get immunity?
Yeah, let's talk about that for a minute. Right.
Is it 4
Where
different-
happened to all that immunity?
Is it 4 different kinds of malaria?
No. Same kind. You can the immunity to malaria. It's a conundrum of multiple.
Is it also a problem?
It's a big problem. It's an issue. So it means that malarial immunity, although it exists, is short-lived. And what does that say?
Short-lived. Would you say short-lived?
I'd say short-lived, but
Lived?
I've heard the different pronunciation by Dickson several times.
Knowles eye and falciparum. I'm sticking to those.
I do think Knowles eye is correct. So
Yeah. So why do you think, Daniel, the B-cell biologist, why do you think immunity to malaria is so short-lived or lived? Of such a short duration.
no. Well, it is. I mean, that is one of the problems.
So what's going on there?
And you see, well, first off, as Dickson was mentioning, this cerebral malaria is disproportionately affects young children. And years back, that was one of my research projects, was looking at cerebral malaria in little kids from Tanzania under the age of 5. Wow. And that is unfortunately, under 5 is when you're seeing most of these cases of cerebral malaria and deaths. And the deaths can be as high as 10% in that population. And the other problem we see, and this would be a travel medicine problem, people leave an endemic area. They're living here in the States. They decide they're going to head back. They're not worried about malaria because, as Dickson points out, hundreds of millions of people can get it, but not that many people die relative to the number that get it. Now they go back and they get really sick. And it is because whatever immunity we develop is short-lived. And the understanding of that immunity is a challenge. The best advance we have in vaccination is not understanding a specific target, but actually taking full sporozoites and injecting them. Here's the whole thing.
Irradiated.
So there's a company, right, that's trying to grow up enough of these. And so it's very crude. It's like where we were with the viruses. Here's a whole bunch of killed stuff.
But
we don't know what you're responding to.
It's not likely to be enduring, just
Actually,
like
it's not
natural
even killed.
infection.
No,
It's
right?
irradiated.
And even in the early studies looking at that, it is not a long-lived. It does not create long-lived immunity, not a long-lived T cell or a long-lived B cell response.
Right. So this age group that you mentioned, that's fantastic to point that out, because almost nobody dies of malaria of age 0 to, say, 6 months. Mm-hmm. It's after that they start to die. So,
they get it, though, at 0 to 6 months, right?
Yeah, but can you guess why that might be, Vincent?
No immune system.
No, that's not the answer, though.
They have antibodies from their mother.
Bingo.
But we just said that
Bingo.
immunity isn't long-lived.
It's not, but there's enough transfer of immunity if the mother has an active case of it or has just gotten over a case of malaria. She can pass that on to her offspring, and they benefit from that. But then there's this null zone that occurs after the antibody's half-life. It's decreased below the clinical horizon of protection. And before they acquire immunity on their own because their immune system is immature, that's the group that really dies the most from this infection.
Do you know why from 0 to 6 months you don't see very much immunopathology, Dickson?
Tell us.
Well,
Tell us.
the same reason the answer I gave you before.
Okay,
because they don't have an immune system
their
yet.
immune system is they're
So
poorly
they're
developed.
relatively
That's
free of immunopathology.
true. Right.
Right.
So there's no innocent bystander effects either.
Innocent bystander. What are you talking about?
Yeah, well, a lot of times when you react to something and you're going to kill it, you also kill a lot of healthy tissue at the same time.
Well, it's not an innocent
Because
bystander.
I would call that innocent
I don't
bystander.
think that's a good name. I don't like that.
But you can find
It's
it.
immunopathology.
It's but it's because
I never understood what they were talking about because it doesn't make a lot of sense. You're not an innocent
you could also have
bystander.
basement membrane disease with this infection because of the antigen antibody complexes that develop.
Should we move on?
We should.
I hate to move on from
We
malaria
have
because
so
it's
much more to talk about.
such a great disease.
That's
but
true.
as we move into our discussion, our article is actually about malaria, so we
Yes,
really
it
don't
is.
even have to leave malaria behind.
That's
It
right.
is. It
That's
is. It's a really an interesting article, which
And as we move on, I'm going to throw the last thing about this case, and I'm going to add a complexity to why do children under the age of 6 months not die of malaria. And it has to do with a little feature in our patient's background. Do you remember what he had a family history of? He had a family history of a certain blood
Thalassemia?
disorder.
Thalassemia. Yeah.
And is thalassemia protective to malaria? And it is. And what is the mechanism? Well, it's complicated, right? I said that last time. But I'm going to throw a little complexity there. Is fetal hemoglobin is very protective to malarial infection.
Look at
And
that.
people with thalassemia have a slower clearance of their fetal
Right.
hemoglobin. So because of that slower clearance, we believe that the thalassemias are protecting us during those first 6 months.
Yep.
Now, other hemoglobinopathies like sickle cell that we're more familiar with,
that's protective
and
as well.
maybe hemoglobin E, they may have intrinsic protective properties.
Sure.
But we think the mechanism for the thalassemias we're in life, which is when we're so worried about this, is this delayed clearance of hemoglobin F.
So fetal hemoglobin is made in the liver. Is that correct?
Sure.
I think it is. Not in the bone marrow. It's
I'm not
actually
sure. I'm
synthesized
not
in the
sure
liver.
where we make that.
The liver. I think it's coming from the liver.
Would you like to know?
I would like to know that, actually. I thought I'd pick that up as a little gee whiz fact along the way of my life.
No, I don't know where we make that. I was assuming there was a little red cell factory where you're making the red cell in the marrow as you're putting it in. But,
No, I think fetal hemoglobin is liver generated.
so Dickson will let us know.
No, Vincent will be the verifier of this fact.
Okay.
Factoid. I know the pediatricians out there that are doing research are laughing like crazy right now.
I don't know. It's not actually saying here. In
Okay,
the first
we'll
few
get that
sentences.
answered for you.
We'll try to get there.
Right. So, how do we begin this article?
Well, I found this interesting because it deals with wildlife again,
Indeed.
and we did a water buffalo
We did.
article,
We did.
and so this is quite. I mean, these are hard experiments to do
They're incredibly difficult.
in wildlife. So this one is in great reed warblers.
Exactly.
Had you ever heard of such a?
Well, I've heard of warblers, but I didn't know this particular one. This is a breeding bird that breeds in Sweden,
Yeah.
but lives its life as an adult in Africa after they finish migrating.
That's a good life.
Not bad.
Grew up in Sweden with that. So the name of the paper is Hidden Costs of Infection. Chronic Malaria Accelerates Telomere Degradation and Senescence in Wild Birds. I found the hypothesis interesting. So we talk about this a lot. What's the cost of disease, right? In terms of evolution.
Right.
So when obviously acute infections that make you sick and you may recover. In an animal, anyway, this is what they're interested in. Could cause a problem. It makes you slower. You can't react, and the lion eats you, right?
Right.
So they're bad. But the question here is, what about a chronic infection, particularly one that doesn't have any clinical signs?
That's right.
And I started thinking about human chronic infections. Of course, in humans, we don't have much selection against sick people, right? Because we care for them.
Right.
Although we do have... all of us have a chronic herpes simplex infection. And I wonder if we were hunter-gatherers, if that would cause a problem in us.
Right. So the other concept that you probably would have mentioned if I had given you the chance is that once you've reached your reproductive age and you've reproduced, so to speak, everything else after that is sort of a bonus to your own life, but it really has very little evolutionary significance in terms of passing the genes on, right?
Exactly.
So why should you live past the age of 40?
Exactly, unless somehow you can help other people live longer.
Well,
but there's no altruism in nature, right, Dickson?
No, that's not true. There's a lot of cooperativity.
Some people
tons of cooperativity.
What's the selective force for altruism? There isn't any.
No, yeah, there is.
Well, that's a big discussion, actually, but it's not this one.
There's some real good ecological studies.
This is a different story.
That's true.
Who
but for humans, there's a huge...
Did you pick this paper or Daniel? Who picked it?
I did.
You did.
Did I pick this paper? Yeah, I did. And then you looked it up and sent it to me.
I think you picked the mention of the paper. And
right.
I actually got
exactly.
the paper.
Yeah, Dickson gets the fluff, and then you get the hard and real stuff.
This works out well.
Exactly right.
The authors are Ashgar, Hazelquist, Hansen, Jedidev, Westerdahl, and Bench. They're from Lund University and the Karolinska Institute and the Bulgarian Academy of Sciences in Bulgaria, Sofia, Bulgaria. Why did you like this, Dickson?
I've never heard of a parasitic infection affecting telomere length.
Have you heard of malaria in birds?
Of course.
What a setup, huh?
Thank you for that, because the reason why we know that it's a vector-borne infection was because of Sir Ronald Ross, while he was in India, working with bird malaria. And he looked at canaries, basically.
And that would be Plasmodium avium?
Well, there are many species of that. We'd have to look up the one. But the mosquito that he discovered that transmits it was a culisine mosquito. So that was a sort of a red herring. It threw off looking for the right mosquito species for human malaria because it turned out not to be the case.
So here they say the species, the malaria that infects these birds are both Plasmodium and Hemoproteus.
Right.
What is
it's another genus
of malaria, really?
Yeah. Yep.
And it does not infect people, obviously.
No, it does not.
Why do they call it Hemoproteus?
I guess to distinguish it from the Plasmodium group, because the Plasmodium group have certain characteristics that group them all together into the Apicomplexa.
So apparently they had all this data stored away.
Right. I made notes on why they call it Hemoproteus.
What is the
so
reason?
hemo, right, meaning blood.
Yeah.
And then Proteus. Proteus is actually, I was reading this paper with my kids around who are very into the ancient gods. And Proteus was actually one of these ancient sea gods who could change shape. And so it's this intra-red cell, intra-erythrocytic protozoa that changes shape of the red blood cells. Like we talked about with O Valley, where it got its name by changing the shape. So this is another apicomplexa, like malaria, that affects these birds.
So what's unique about red cells in birds?
They have no nuclei.
The other way.
They do have nuclei.
They have nuclei.
Right. I know that because when I was a student, I was doing a study of chicken erythrocytes, and they had nuclei.
Exactly.
Yeah.
What's going on there?
Don't know.
So do they have a metabolism?
Dickson, do you think dinosaurs' red blood cells had nuclei?
That's a great question. Let's go find out.
I'm going with yes.
The new Jurassic movie is going to come out soon.
Yes would be a good answer.
I know yes would be a great answer.
So they had apparently they had been studying these birds for a long time.
Sure.
They have identified 17 different malaria parasite lineages in this study
That's
population.
amazing. That is amazing.
So they have a study population. These people, every year, they study these birds, and they take samples, and they see what's in them.
Right.
And they could then ask, is there a problem in having...
Exactly.
So apparently they can be long-term infected, chronic infected, and they're not sick in any apparent way, right?
But tell us what you think you mean by long-term, because the average age of a bird is not that long, right?
How long does a bird live? That's
well, the ones that are not infected and therefore are not shortened in terms of their lifespan live about 2.5 years.
That's not very long, is it?
It's not. I
mean, like a mouse,
I guess for a bird it must pass very slowly. But for humans, 2.5 years is like you blink your eyes and you're dead. That's ridiculous.
They say here in wild bird population. Well, first of all, the thesis, right? Are there any effects of chronic infections? They say such data are scarce for animals.
Right. Correct.
Which you can understand because it's
Yes.
hard to do, and also probably hard to get funded because everyone's interested in human
wasting disease is one of the exceptions too, right?
Only because we would like to prevent it infecting people. Exactly. Right? I mean, that's the ultimate.
Exactly. By the way, some birds live long lives. These warblers are songbirds, and they live short lives. So there are.
How long is long?
Well, penguins live long lives. I think they're fairly old birds, like 20 years old, something like that. And
Don't get too close to the mic. You're peaking. Right?
I need one of those guards like
I know.
that prevents you from getting close.
Well, don't get close. Make an effort.
I'm trying not to.
No, I will get you one of these. So, because you're making really... You're doing what we call...
Are you popping?
I believe I'm
popping.
But even beyond that...
I'm scruffing my beard up.
So you see these waveforms? When they hit the top, that's called peaking, and you never can recover that again. It's gone. So what was above is gone.
I have a feeling that
clipping is the name.
You're going to edit this whole thing.
I can't edit it, no.
You can't edit it. No.
Now, are there any 3-letter acronyms for this phenomenon? I realize we've gotten quite far with no 3-letter acronyms. What has happened?
Exactly.
So they have these, they've been studying this. They have blood samples, and they can look for infection. They use PCR. Right? And they say low-level chronic malaria appears to have no direct short-term costs. How would they know that? What are they measuring?
Dead birds.
Oh, dead birds, huh?
Yeah.
So, but all the ones they're measuring are alive. So
or
how would you?
Or the drop in reproductive index.
Well, no, but I guess that's in contrast to what they're actually then going to go on to say. I underlined that sentence, and I was, what are you looking at?
Oh, I see what they say here. There's no difference in activity patterns, either in song rate in males.
Right.
So that's one.
Yeah.
Oh my gosh. Song rate
Nestling
in males.
feeding rate in females was the other.
The percent of the time singing every 15 minutes. Dickson, do you sing a lot every 15 minutes?
Not every 15 minutes.
That's how we would measure your fitness. Yes, and the females.
Spend the majority, of the time singing, right? 60% of the time these birds are singing.
Happy.
And nesting feeding. So nestling feeding rate. So those two parameters are what make them say there's no short-term effects of being chronically infected.
Right. But
they said they have significantly shorter lifespans.
Almost
so the
a
mean
full year different.
Infected is 1.69 years, and uninfected is 2.53 years.
That's
and they have fewer
remarkable.
Fledglings.
So
what is a fledgling, Dickson?
A fledgling is a bird that's hatched and then succeeds in leaving the nest and becoming an adult.
So I'm not a fledgling.
Not anymore. But you were at one point.
So they're very specific words.
Absolutely.
Do you know what a male swan is?
I do.
Tell me.
It's what do you mean?
Is it a cygnet or something?
Oh, you mean does it have another name?
Yeah, like a male.
I think cygnet's the right word.
What's good for the goose is good for the gander.
And then the female pig is the sow, and then there's
they have
all kinds of names.
Animals have different names for the male.
I
and
love
females.
that. I love all
This
that
is
stuff.
true.
So fledgling is pretty cool. I like that very much.
Okay, great.
So they have shorter lifespans and they make fewer offspring, which they could tell in their
study. They don't make
study.
fewer offspring. They have fewer fledglings. The number of offspring is exactly the same, but the number of successful offsprings that enter into the adulthood is reduced.
That's a very important point.
Extremely important.
Fewer fledglings.
Yes.
Yeah, you're right.
Yes.
I'm trying to see if they actually measured the number of offsprings to see.
The number of eggs are the same, but the number of successful rearings is reduced.
So that they say right away that this disease, chronic avian malaria, with seemingly negligible short-term costs.
Yeah.
At least for singing.
So why are they going to die a year ahead of schedule? That's probably what they wondered, isn't it?
They wondered, and they got into the telomeres. Is it telomeres or telomeres? I would say telomeres.
I'm sure you're going to tell me.
Telomeres. Yeah, Dickson, do you know what a telomere is?
I do.
Do you want to explain it? Or are you going to screw it up?
I won't screw it up too badly. It's a series of repeated nucleotide sequences which caps off each chromosome.
Why do we need these?
I guess, why do we need them, or what do they do, you mean?
It's the same question.
There's no such thing as a why question.
What's their function?
Right. What is their function? That's a great question. And because every time a cell divides, one of the repeated cap sequences is cut off from the cell. And so they've only got so many of these, and they don't replace them. And eventually, when they only have one left and they divide, the cell dies.
You know what happens, Dickson, if you make an enzyme all the time in a cell that keeps them long? It's called telomerase.
You immortalize the cell.
You do. It's one way to immortalize a cell. Yeah, and also to cause cancer.
You know why I know this? Because I published Judy Sulzberger's book called Younger.
Really?
And it was all about that concept of telomere lengthening and telomere shortening.
So are your telomeres pretty short, Dickson?
Very short. They're about to disappear, and then I'll just, poof, one day I just won't show up.
They also say that when you stress cells, the telomeres become short. And what happens when they do become short, Dickson?
Well, I guess they are limited in terms of the number of division cycles.
They die. The cells die, apparently.
So it triggers the initiation of programmed cell death.
Right.
Which everyone knows is also known as apoptosis.
Apoptosis.
Apoptosis?
It's not apop.
Apoptosis.
Listen, I made this mistake with Jane Flint, and she corrected me.
This is a baseball term. This means you only hit line drives.
No. Look, you look at the Greek words PTER. Do you pronounce them pater? No, they're teroptic.
Teroptic. No, that's
right.
true.
So it's apoptosis, not apoptosis.
Well, I've heard it referred to as apoptosis.
I know you've heard it referred to.
I didn't say that.
And you've heard Daniel call it something else, which isn't right, but that doesn't mean it's right.
We could go on like this for a long time, folks, but we're not going to.
So they looked at telomere length, right?
They did.
By PCR.
How can you do that for every cell? I mean, how do they know that the telomere is short?
You're obviously getting an average per sample.
Yeah, but what are they sampling?
Blood samples.
Just blood.
Taken when 8 to 10 days old, and then each year throughout their lives when breeding.
So they're measuring white cells and red cells, right? Because red cells have nuclei.
Yeah, they do both. They just take blood samples.
They don't know how to do them separately.
They probably spin out the cells and do PCR on the pellet, right? Or maybe they do total blood. You can do it. It's sensitive enough.
I guess.
It's fine. They found telomere length in the warblers decreased with age, and more steeply so in infected compared with uninfected birds.
Right. Well,
and
whoa.
Isn't that clear?
Red blood cells of an avian species do not divide. Even though they have nuclei, they do not divide.
Well,
so
maybe
you'll have to explain to me
do you think they
how they did this study on red cells that have nuclei, but the red cells don't divide. So how can the telomeres get short?
Did they do Buffy coat, Dickson?
I presume they did, actually.
Right. Well, it doesn't say in the actual discourse.
But red cells wouldn't be the one to look at because
all they say is blood, actually.
But they're not dividing cells. They have to look at dividing cells.
They call them blood samples. That's a really specific term, blood samples.
It has to be the white cells in there, though, that they're looking at.
Probably,
yes. Yeah, so those are non-infected cells. Because the malaria parasites only infect red cells. This has to be clear to the listener.
That's a very important point.
I think it is.
Because the implication would be that the cell that's infected is getting telomere shortened, but it's not.
That's not the case.
So if these animals with chronic malaria have shorter telomeres,
right,
or that decreases at a steeper rate,
right,
but those are not the cells that are infected.
It's a secondary effect.
What's happening?
I thought I asked that question.
Are the infected red blood cells producing something which is toxic?
Or the parasite themselves, right? Like their waste products, for instance.
Yeah, it could be.
Because they do reproduce.
Degree of telomere loss is related to the severity of infection.
Now, what I also find interesting is that you asked a question before about immunity. These are long-term chronic infections. How come these birds don't kill it off? But they don't. They remain infected, which is interesting because In
Well, it's only people
they
you
do
said
fight
they-
it off to the
5
point
years.
of in falciparum infections, you can fight them off, and there's
I
no
know, but the
parasites
Vivax
left.
infection that we just talked about.
Yeah, but that's different.
So the Vivax in Valley will stay dormant, and while it's dormant, we can
Yeah,
do nothing
can't get
about
rid
it.
of
But
it.
once it becomes active, then
We fight it
most
off.
people will either die or
Right.
you
I don't know what the life cycle of this is in birds. Do you know? Are you
Same.
familiar?
It's about the same.
That's what? Vivax
As
or
most other
falciparum?
yeah, that's right.
Falciparums?
That's correct.
I mean, that's a controversy, but we can bring it up. The thought is, even though we like to blame gorillas and the great apes for everything, and they may actually be involved in Vivax Valley transmission, there's an idea that we may have gotten falciparum from
Birds. That's right.
Which is in the time of Ebola we can be frightened because these birds,
I know.
they're heading down to Africa, they're hanging out,
This
they're
is
getting
true.
malaria,
And you know
and
what's-
then they're flying up to Europe
You know what's-
with malaria in
Do
their
you know
blood.
Do you know what's weirder even than that about falciparum malaria? It has plant genes. Plant genes.
For what?
There's a whole sequence of plant genes inside the falciparum genome project. It's been
So
sequenced.
they also infected some animals. All right?
Right.
They could actually, they have them,
Right.
I don't know if they're in the lab, I guess they are.
Yeah.
Experimentally infected great reed warblers experienced a higher rate of telomere loss than uninfected control birds over 9 to 10 weeks. So I guess it's in the lab, it's a controlled infection. You see the same thing.
Yeah.
Now here's the interesting part. You remember this? Malaria-infected
Wait a
mothers.
minute, I have to wake up.
Malaria-infected mothers produce offspring
Yeah,
with,
yeah.
on average, shorter telomeres
This is bizarre.
in comparison to the offspring
It's bizarre.
of uninfected mothers.
This means Lamarck was right.
How can? So why would?
Exactly. So wait a minute. Maybe we don't understand the true mechanism of how to shorten telomeres.
Well, we do.
We're
do
blaming
understand.
it on the parasite, but maybe there's some generalist phenomenon going on here, like raised body temperature or increased metabolic rate, because they also mentioned in their discussion that other diseases can also result in shortened telomeres.
Here's another corollary. So the control for
Right.
this experiment, as uninfected mothers become older,
Yeah.
they produce chicks with successively longer telomeres.
I like it.
Unless they're infected.
Unless they're infected.
that
Unless
is
they're
great.
infected, then it's the other way.
Why would they
That's
do that?
remarkable. I mean, it's not a why question. How do they do that?
How? Well, how
What's
we,
the function?
And what would be the result evolutionarily of producing older and older and older offspring that we could live longer? That's what I meant to
Well,
say.
that's it. As they get longer telomeres, they can presumably live longer,
What
right?
one would
But
assume.
why is this? As the mother is making more, why is it increasing? It does not make sense. I don't know what the function of that is.
The ultimate way to look at this, though, is that even if a bird could live longer, it probably won't because it'll encounter some infection that'll shorten its lifespan.
Yeah, Dickson and I did discuss a little bit off mic the issue of, so you are born and you have, your cells all have telomeres of a specific length. You could say it's
Now
sort
he's
of our
calling them telomeres.
What was the telethon?
telomeres.
Telomeres. That's right.
These,
Telomere.
no, actually,
Tilapias.
I was walking the dog last night and I was talking to my kids about this stuff. And little Barnaby at 9, when I asked him what they were, aren't those bumpy things on chromosomes?
Bumpy things.
Get out of here.
My 9-year-old.
Your 9-year-old
And,
kid said that?
Well,
Well-
bumpy. He's probably thinking of nucleosomes.
You know, you've been
right?
reading him too many bedtime stories.
Yes, I should have corrected him. Barnaby, I think you're confusing your telomeres with your nucleosomes.
That's right. Oh, sorry, Dad.
No, I had lunch and dinner with one of the gentlemen who won the Nobel Prize for his work on telomeres.
Who's that?
Jack Svotak.
Shostak.
Svotak. Yeah, see, I can't
Jack
pronounce anything.
Shostak, yeah.
I'll just pause and you can finish my.
Go ahead.
He was very nice. Jack did not correct me.
Really? How did he pronounce
Vince
it?
can be the intron on this one.
No, I was the one who mispronounced it, and he was very nice to me.
He never
didn't say a
said
word.
the word himself, right?
No. But,
You know what go ahead.
but no, so we talked about these. So you're born and you have these telomeres on the end of your chromosomes. And, every day you're worried because I wake up and, oh my telomeres are a little shorter than they were yesterday. I only have
in
so
some
much time
tissues.
left.
Only in some
But there are
tissues.
3 areas where the cells actually elongate their telomeres. It's the fountain of youth.
Huh.
One, as we're seeing here, is down in the, and you got this correctly. I was trying to call them like malaria reproductive forms, but our reproductive cells will actually lengthen their telomeres. Because,
the
gametes. You've got a 30-year-old woman, let's say, and she's going to have a child. You don't want to give your daughter, your son, short telomeres. You want to lengthen them so they can start off with the long. So you have telomerase, the lengthening active there. You also have it active in our stem cells. Our stem cells can't get older and die.
What's the enzyme called that shortens them?
No, they just shorten during division.
Yeah, there's an inactive.
During DNA polymerization,
every time you divide, they get a little shorter.
It stops at the beginning of the next repeat.
Well, it doesn't go all the way to the end. That's the problem.
See.
But
and that's part of why you need these.
Telomerase restores it.
Yeah. And then our B cells. When our B cells go to the germinal center, and now you want to have these antibody-producing cells to live for a long time, they actually get their telomeres lengthened.
Get out of here.
Yeah.
So there's something that actually is doing that in the thymus?
When the bone marrow is in the germinal center, and that's going to be mainly spleen and a few other germinal centers, there's the active telomerase enzyme which lengthens those.
My goodness.
Reverse transcriptase telomerase is actually the enzyme that lengthens them after each cell division. So in addition to Jack Szostak, Elizabeth Blackburn and Carol Greider also won the Nobel Prize for discovering telomerase in 2009. You want to know where they're from?
Sure.
Liz Blackburn was from UCSF.
Okay.
Berkeley, sorry.
Oops.
And UCSF. Oh, she's been all over. Berkeley, UCSF, Yale, Salk. She was actually born in Tasmania.
Really?
And
That's Tasmania,
Jack Szostak is from Yale.
Is that how you...
I'm pretty sure.
And
Carol Greider has been at Cold Spring Harbor and Johns Hopkins.
Oh.
Not sure where she was. She discovered the enzyme in 1984 when she was a graduate student with Elizabeth Blackburn in Berkeley.
Wow.
How'd you like that?
Very nice.
Cool. Anyway, so now we have this interesting fact that these
we're
birds,
stuck. The warblers are warbling.
Malaria. How does malaria make them shorter? These telomeres.
Right.
Any other questions?
I like the question of, so here's birds, and I give the other people in my lab a hard time. Are we studying people? And now that we know this in birds,
we should look
does
at
this translate into people? People who are living in an area with a lot of malaria, they're getting it, a lot of people every single year, they get this low level. Do they get shorter telomeres?
Well, and then superimposed on that, we have good statistics with regards to populations and their cross-sectional ages. Is there a correlation?
We must know in human populations if malaria impacts longevity. That has to be known.
The saying that I've always heard at tropical medicine meetings is that if you can live past the age of 5, you're going to live as long as anybody else in the world. So there are tons of childhood diseases.
Sounds like an old wives' tale.
No, it's their observation. There are lots of old people out there in the tropics.
I'm not sure. Is old wives' tale still politically correct? Can you say that anymore?
By the way. I did not say that.
I'm just checking.
I want it clear for the record.
I say a lot of politically. I
also am wondering if
countries.
your mom has active malaria. And are you born with shorter telomeres? Right. Is this something that gets passed on to the next generation, like we're seeing in
very
the
interesting question.
So malaria in birds, I believe, replicates in not only in the red cells in the circulation, but I think also in the bone marrow. Because Dr. Traeger, William Traeger at Rockefeller University, who I came to know very well because I was a postdoc over there, worked on Plasmodium lowfuri.
Yeah.
And Plasmodium lowfuri infects ducks.
Right.
And the unique thing about that is that if you inject all the ducks with the same stage, you can, in fact, have your all ducks in a row because it all replicates at the same rate. You get a synchronized infection, and they will all die on day 12. If you don't come into work on day 11 to collect the blood for those stages, he was trying to grow them in vitro, then you come in on day 12 and they're all, duck soup, basically. So it was a fascinating model to work with, and he spent a lot of time explaining it to us all and reveled us in his Plasmodium lowfuri stories. So I don't think it's Lowfurian's bird population, but there's tons of them out there, tons of them. There's a big book on malarial species by Garnham that I have on my bookshelf, and it's just page after page after page of these blood smear pictures and names that you'd never heard of before. Plasmodium gallinaceum
Yep.
is the one I was trying to remember, and that's the one that infects birds that
Okay.
Ronald Ross worked on. That got him his Nobel Prize. Speaking of Nobel Prizes.
Let's move on to the next case.
We're into
Should we
the
move on
case
to the
history.
next
Yeah,
case?
we need to do this.
You got it.
All right. Well, I was enjoying this paper so much. I could have spent even more time. And the last thing I was worried about is the interesting thing about, when you're in Europe and you go south for the summer, you go to Africa. We're all worried about things, people coming in, bringing things in, but it looks like these birds are just traveling with impunity all over the place with these horrible diseases.
I mean, this is how West Nile got to where it got to, right? Because that's where it comes from too, Central Africa.
All right. Well, I have a wonderful case.
Regale
I'm going to
us,
say it's
please.
a wonderful case. And as I go through these, I was saying initially I want to start off with a theme where if you've been a loyal TWiP listener, you'll go through this, and then the history is going to give it to you. If it's and this time, what I want to do is I want to engage Vincent maybe a little bit more in asking the questions.
I've just dropped out of the conversation.
No.
He can be involved, but I am going to.
AIDS.
But I am going to say yes. Oh, how did he know?
Superimposed with polio.
Yes. All right, let me start off.
Please.
And the other thing I would say, I want you to think, I want you to ask a lot of questions, but don't mention any specific pathogens until maybe the very end. We'll see if that's even allowed today. We start off with history of present illness. This is the story. And a 56-year-old gentleman comes into the clinic in New York with concerns about his leg. The backstory, actually, I was in the ER, and I'm a pretty friendly guy, and so they see me there, and they say, "Hey, Dan, come take a look at this." And I say, "All right, send him to clinic." So I see him the next day. So there was that. I see him in the ER, and I decide he can be seen in the clinic.
What did you look at? His leg?
I looked at his leg. That's what they wanted me to say. And he was concerned about his legs. And now he's brought in by his concerned son and daughter-in-law who live in Western Long Island. And the man has come to visit him, and they are upset at the appearance of, this boy's father's right leg. Okay?
That's really huge, right?
Vincent is already gesticulating beyond belief.
This is when you're
Just
like,
one
this
right
is like
leg?
you're playing that thing with your brothers, and you want to change the answer at the end because they're getting too close.
That's right.
No, we're
I see
stuck
something
with the
red.
answer. So they're concerned about his right lower leg, and he reports that he's had swelling of the right leg for several years, and that now there's an open, non-healing wound.
Oh, dear.
He doesn't have any fever. He's got no constitutional symptoms.
Uh-oh.
And what does that? What does that mean? No constitutional symptoms.
He feels okay generally,
Yeah.
except for his leg.
Exactly. No fever, no chills. He's not clattering his teeth, things like
Wouldn't
that.
you say systemic symptoms instead of constitutional?
You could say that too.
It would work. Okay.
Yeah, you could do that. Constitutional is just a big fancy word, right?
It is, well, it's,
Well, for me, it's a big
it
fancy
becomes
word.
political. It
These
sounds
are-
political to me.
Medical diversions to keep the patient from actually understanding
Yes.
what that's going on.
Now, Vincent, what would you like to know?
Where was the open wound? In the ankle or on the upper thigh?
It's on the medial aspect of the right leg, so that's towards the midline, and it's just above the ankle. I'm going to show you a picture, and then
I got
and
it
this is
already.
actually
no
a picture.
specific pathogens,
All right.
remember? You have to
And,
lead the audience.
We're gonna let the audience generate a
Okay.
differential. Okay,
But
so
where's he been?
oh, that's a great question.
That is the question, actually.
He was born and grew up in Georgetown. Now you may want to ask.
That's Washington,
You may
D.C.?
want to ask which Georgetown.
Where is
No, that's
George?
got to be Africa, right?
It's a coastal town in Guyana, so the northern part of
nice.
South America.
Not
So
Africa.
grew up in Georgetown, which is a coastal town in Guyana. But he reports that he traveled often to the U.S. for business. I tried to ask him what business he was in, but he really didn't want to answer, which I thought was curious. So he's involved in some sort of business that involved lots of travel to the U.S.
Did you look at this open wound, Daniel?
I did.
And was there anything? Was it oozing?
So you want to, but remember, we always ask a lot more questions before we actually touch and examine them.
Okay. So he's been there, and has he? What has he been eating and drinking?
He doesn't report anything remarkable. We ask him about, unpasteurized cheeses, things like that, and he says no.
So he didn't filter the water before he drank it.
He does not report any specific actions that would yes. Yeah, he did not drink any water through a filter straw or anything like that.
All right. Well, I already know what it is. I don't have any more questions. What about you, Dickson?
The prosecution rests this case, I see. No, I've got tons of other questions. Like how big a lesion is this?
Okay. So the lesion is 5 by 3 centimeters.
Not centimeters.
No, centimeters. And actually, that was the way one of our professors at NYU pronounced it, and that's the way we could recognize one another. Oh, you also say centimeters.
Well, my wife's mother was a nurse who trained in Canada, and she called it centimeters also.
Centimeters.
Yeah.
So I say 5 by 3. It almost seems a little bit larger, right? This is if you look at the palm of your hand,
right?
It's a little smaller than the palm of his hand. On the inside of the right leg, a little bit above the ankle bone.
What about anything else about the leg that might be unusual? Like, is it indurated or is it tender to the touch above that lesion, perhaps moving towards the knee?
Well, I wrote out a description here.
Yeah, please do.
And I say right leg with significant increase in girth compared to the left. So it is, if you put a measuring tape, or it's actually the right leg is larger. There is, we say, brawny edema.
Brawny edema.
Brawny edema.
Oh my. That's very florid language for a physician.
And it's important in the description, right? When you're going to be presenting this case, discussing with colleagues, you're not going to usher everybody in to see this poor gentleman.
Right. Is the edema area that's brawny also tender to the touch?
No, it is not. Now, the brawny edema, brawny is somewhat different than we describe pitting edema.
Right.
And we actually do this where you put pressure with your thumb and you see if you can leave an indent. That would be
rebound edema.
Pitting edema.
That's right.
And this is you push. You're not going in with the thumb. This is just a swollen, thickened,
solidly cellulitic.
And the skin actually looks cobblestoned, hyperkeratotic.
Oh my.
Okay.
But Vincent is nonplussed at this point. He's not thinking right
and
now.
It almost looks like
they're get on with it. I know
almost
what
looks
this
like
is.
there are loose plaques toward the. So maybe this is important for our listeners to get here. This is not a normal healthy leg with a lesion.
Yeah.
This is a leg with chronic changes.
It's not a carbuncle
with
ruptured
with an open.
How long has he had this leg issue?
Another great question.
Right.
He's had this leg issue for many years.
Mm-hmm.
Many years.
Many years.
And only recently has he had this open lesion occur, right?
No, the open lesion has been there for months. And according to him, it really hasn't changed very much. He's not that upset about this. And there's no drainage coming. Actually, the wound does not look terrible.
Really? Do you think you know what this is?
Now I'm beginning to wonder, because that's not right. That's not right.
Thank you. No, not for your diagnosis. That's exactly right. So think about Guyana, South America, all of the things that could be down there that could cause a lesion.
Well, you haven't taught me country by country, Dickson. That's the problem. You've taught me parasite by parasite.
Well, there are few, the ones that you know of that would cause a lesion.
Where is Guyana in South America?
It's
Whereabouts?
It's northern South America.
Oh, it's right next to Venezuela.
That's right. So there are a lot of possibilities here. Loads of them.
Really?
Oh, yeah.
All parasites.
Well, many of them.
I think he has AIDS.
You could have a weird fungal infection, for instance. They have a lot of weird fungal infections down there.
But he's had this for a long time, and it's not.
He has. Well, the fungal infections are long-term chronic infections. A lot of them.
And his leg doesn't bother him. It must. That's why he came in, right?
No. It bothers his son.
I see.
His daughter-in-law. They're bothered. They're the ones who bring him in.
And everybody else on the beach.
It doesn't really bother him.
Usually the patient doesn't come in unless it's really bad, right?
Right. This actually doesn't hurt, or it does hurt?
It does not hurt.
Oh.
It does not hurt.
This is a very important
This is a painless.
This is a very, very, very important
painless
clue.
process.
Vincent, this is
That's really important.
not what you thought it was.
Okay.
Because of that one particular thing.
Because it's painless?
Yes.
Painless.
Yes.
It's something secreting a nerve toxin of some sort.
He reports lots of animal exposure. So he lives in a rural part of Georgetown, kind of the outskirts.
How high up on the leg is the swelling? How far up does it
I'm
go?
gonna actually. There's a picture of it, Dickson.
Well, that's too bad. You
and
showed
it
that to me
goes
because I now
and it
know what
goes
it is.
about and it goes
Take a look.
about midway up, halfway up
Midway.
to the knee. Doesn't quite
Midway
make
up
it
to the knee. So you've got a.
And the skin is much darker, right? This is a colored band,
This guy,
Pigmented.
but it's
I can't
also
believe this guy wasn't
really
worried
dark.
about this.
Oh, he wasn't worried.
Oh my
Because
God!
everybody else where he lived has that too.
Well, not everybody else, but he did mention that he's
Yeah.
not the only one in the
He's
town.
not the only one. And it's only one leg.
It's only one leg.
It's one leg.
Is that typical for what you're thinking about?
For me, it is. Yeah, it is, actually.
Okay. Is there anything else we need to know? Is that all you're
I
going to
want
give
see
us?
the blood smear.
Well, you can't get that till next time.
Well, then you have to suggest tests then, right?
No, so our listeners can suggest tests. They can suggest a differential diagnosis. They can even suggest more questions.
All right.
And the last caveat I tell you is our patient says, you realize, Dr. Griffin, I have very limited funds.
What does that mean?
Wait a minute. What's all these trips from Guyana to New York all
and
about?
and no health insurance. So whatever you want to order, I'm going to have to pay for out of pocket. So be judicious in what you do next.
So he had a lot of animal exposure, and I presume he had mosquito bites, right?
Yes.
All right.
Tons.
He also didn't see any no-see-ums, just like our last person.
Okay. Right. That's right.
Well, what I'm thinking of is a real problem in Guyana. So,
Oh, you are, eh?
and what you're thinking of too.
I
Did he take any precautions to avoid mosquito bites?
Such as?
Repellents or long sleeves shirts and long pants and so forth.
No,
He did not.
he did not.
Did not sleep under a bed net, right?
He did not.
Treated with something. Okay. All right. Well, I have enough.
Okay.
You've had enough, or you've got
No, I
enough?
think I know what it is.
So, Dr.
Because
Michael
I was on the
Katz-
wrong. I was on the wrong track,
Yeah,
but I saw the picture. Yeah,
So
that's-
one of the great teachers at this university, Dr. Michael Katz, who was the chairman of pediatrics for many years, and I had a close relationship with, used to give the lecture on malaria, and he used to say that if you go to sleep and you hear a buzzing noise, he says the female Anopheline mosquito doesn't make any noise whatsoever. So if you hear a buzzing noise, you shouldn't worry. He says, but if you don't hear a noise,
you should wake up. Yeah, that makes a lot of sense.
and everybody sort of sat there and said, if you don't, wait a minute.
One more question: how are his
Yes.
genitals? Normal?
That's an excellent question. His genitals were normal.
All right.
But we'll
So
maybe.
this was from the knee down.
Have you noticed that last time he wanted to know about sex, and now he wants to know about the genitals?
Yeah.
But no,
this
they're
is-
actually very. I would say that's a very relevant question in this case.
And elsewhere, he's fine. It's just the leg, right?
So we'd love to see some more
Yes.
questions, but I would like to see a big list of tests. Not too many, though, because he can't afford the big ones.
Well, he's gonna.
You have to do the classic tests.
We have to.
We have
wait
to
till next
evolve
time.
into this.
I think
That's
this is as much as we need. I think everyone's going to get this.
You do?
Yeah.
I don't think that's an easy
Well, they
one.
can't see the picture, but the location. If they just they can look it up. Actually, can find it.
They can look it up.
Look it up.
Isn't,
what are you
isn't,
using,
isn't
Watson?
Google,
Are you
isn't
using
Google
Watson here?
great? No, I think even Google, they've done studies on this. Google has really
They've come on board.
been helpful, particularly when you're looking at diseases that are occurring in other
Yeah, of
parts
course,
of the
of
world.
course.
And that's one of the nice things about parasitology is there are certain things. We talked about babesiosis. If you present a case and here's someone from Nantucket, I think we kind of know what it probably is going to be. And, depending upon where the person is from,
Right.
there's going to be certain diseases in that geographical location.
Sure.
And maybe someone on the planet can have every geographical location memorized and what pathogens exist there.
Sure.
But thanks to computers, thanks to Google, thanks to the CDC and the updated websites, it actually is very helpful to be able to go to these places and get the information which you can then immediately
Yep.
bring to the benefit of the patient.
So would you like to give the classic quote that pathologists are fond of saying with regards to what to think of when you see something?
Yeah, zebras, not horses.
Yeah, well, when you hear hoofbeats,
Yeah, think of zebras, not
no,
horses.
think of horses. Don't think of zebras. That's their initial statement.
Why?
But if you're in tropical diseases and you're in the United States, if you hear hoofbeats, in this case, it
So
may turn out to be a zebra rather than a horse.
Don't think of unusual things.
Exactly
Just
right.
standard.
Exactly
Okay.
right.
I only have a few letters. Let's read them.
Sure.
One is also from Robin, who sent an article.
Maybe we should make Robin a regular.
I bet he'd love to be on the show.
Our foreign correspondent, Robin, writes.
Horizontal transfer of functional nuclear genes between multicellular organisms.
Wow.
In this paper, they demonstrated the transfer of genes encoding chloroplast proteins from an alga to an ascoglossin sea slug.
Remember, I told you that Plasmodium falciparum has plant genes. How do you think they got there?
Well,
Maybe Robin knows.
they said, without doubt, transfer of functional genes is routine between prokaryotes, has occurred between both mitochondria and chloroplasts and the cell nucleus, and DNA has been transferred from endosymbiotic bacteria into insect host cell nuclei. However, no evidence exists for natural transfer of nuclear genes between multicellular organisms.
So what do mosquitoes eat?
Oh,
It's a trick question.
They can eat blood, right?
Trick question.
Females drink blood.
Just the female, though.
Do they eat sap?
They eat cell sap from plants.
So they
That's
have
what they-
endosymbionts to provide amino acids that they don't get from the sap, right?
Maybe. Or maybe they could pick up some chloroplasts by accident. And if they happen to have malaria at the same time, it's possible
are you
that the
speculating?
genome...
Or is that
No, well,
Speculative.
I'm already telling you something. It's a fact that Plasmodium falciparum has plant genes. How did they get there? That's the point.
Where did you read that?
It's already in the genome. They've sequenced the darn thing. I saw a talk on this once where the guy actually painted the mice green. And he had them on the table, and he said, these mice are infected with a Plasmodium falciparum-like protozoan, which,
Well,
if you
it's-
sequence it, you can find plant
It's not
genes in them.
multicellular, right?
It sure is.
So
No, what's not multicellular?
Is Plasmodium.
No, it's not. It's not multicellular.
So-
But the plant is,
yeah, I know, but between multicellular organisms. That's the point here.
or eukaryotes.
Anyway, that's
All right.
the article. So
Okay.
it's cool.
No, it's very.
But
cool.
we find, sequencing technology is now at the point where we pick this up. We see lots and lots of examples. On TWiP last week, we did a cool paper where, so there are a number of bacterial genes that are antibacterial that target the outer membrane of the bacterium. This has been transferred at least 6 times into metazoan species, including ticks, and they could actually show that the product is active in a tick. It's in the salivary gland and in the midgut,
kidding.
and it probably helps keep the levels of Borrelia down,
I'll be.
because these are the Lyme tick, right? It's very cool. We're going to see more and more of this.
Hot darn.
Steve writes, one for Dickson. Dolphins can carry a parasite in the part of their blubber most likely to be bitten by sharks because part of the life cycle is in a shark.
That's remarkable.
And he
That is
sent
great.
an article from Dorset Wildlife Trust, which talks about this.
Huh.
Yeah, 2 dead dolphins were reported washed up along the Dorset coast.
Wow.
They had what experts believe to be a large shark bite taken from the rear. Interestingly, dolphins often carry a parasite in their blubber, which requires a shark host for part of its life cycle. Do you know about this? That's an interesting one.
Did not know
They don't
about
give the name of it. The parasite lives in the part of the dolphin most likely to be eaten by a shark to ensure that's a human interpretation of what's going on.
It's a little bit pushy on the evolutionary
What would you call
biology
that?
there.
What would you call that when you humanize things to make
Anthropomorphize.
it make sense, right?
Yeah, anthropomorphic.
Keep them coming, and thanks for your kind reading of my letter on TWiP. That was a nice surprise. Steve is in England. Marcia writes, I'm sure others have pointed this out to you already, but this was an interesting article. And this was we did talk about this a while ago.
Yeah.
And probably, Daniel, this guy had headaches for 4 years, and he had a tapeworm living in his brain. Chinese fellow, and they did MRI, and they saw, and it actually was moving around the man's head.
Yes.
I could actually put it on the screen for you to see.
Oh, yeah.
See, look at the MRI. It's moving around.
Yeah.
What technology we have. Man went to see his doctor after. Hey, this could be a case history for us, right?
It could be.
All right. This is cool. I could put the email up here.
Yeah.
I can't see it because the microphone's in the way. On another more trivial... I have to put it here. I can't see. On another more trivial subject, why is the title of the show This Week in Parasitism and not This Week in Parasitology? When I was growing up, my mother worked in a hospital lab, and that department was called Parasitology.
Right.
One of your other podcasts is called This Week in Virology. Just wondering why the different nomenclature. Thanks for all the enjoyable episodes. It's fun to learn new stuff.
Good.
All right, so Dickson and I had this big argument the other day about this.
Not a big argument. It was a small disagreement.
A knockdown, drag it out.
Oh, yeah.
Because I said it was your idea. You said it has to be called parasitism because we're going to be talking about the concept of parasitism, not just individual parasites. So it's not parasitology, it's parasitism.
My long-term
I
memory
swear
sucks.
you told me that. I would never have come up with something like that.
Trust me on this one. I forgot. I don't remember how that name ever got stuck.
That's it. I'm telling you. You have to trust me.
I can't imagine myself actually agreeing to that.
You suggested it. You didn't agree to it. You suggested it.
Parasitism. No, I would have said parasitology, but that's okay.
You said parasitism. That's why we named it. I would never have named. This was your show.
I'm not doubting your veracity. I said I'm not doubting your veracity.
Anyway, Marcia, that's why. Because Dickson's idea was we were going to focus on the concept of parasitism. That is one organism benefiting in some way from another.
I do feel like you guys discussed this, right? Back in one of the
Absolutely.
earlier...
Absolutely.
Yeah, it was actually on one of the recordings.
And I have to find it. I have to go back and listen because I'm going to show you that it was your idea.
Fine.
Do we think it was a bad idea?
No, I think it's a fine idea. I don't but you know, when most people cite the show, they call it This Week in Parasitology because they assume that's what it should be. Yeah. All right, one more. Danny writes, hello, TWiP Musketeers. Yeah.
I like that.
There's the three of us.
Wow. I like that.
I love the new case of the week. Thank you for this most interesting addition to your educational empire. I'm emailing today about lizards and Lyme disease. I've recently learned that some lizards that are a host for tick nymphs actually clear the nymph of the Lyme spirochete.
Wow.
Nature amazes me. I've done a literature search looking for information on the biochemical basis for this phenomenon with no luck. What do you guys know about this? Is this phenomenon useful for trying to manage Lyme?
I bet I know.
The weather is 16 degrees Celsius in sunny Cupertino, but I prefer the rain. Thank you for the
They would because they haven't had any.
podcast. Cupertino.
Yeah.
I worship Cupertino.
You do, eh?
Yeah, I love my Apple computers.
Right.
You know anything about this?
I don't know anything about it, but I think there are naturally occurring antimicrobial agents known as meganins, which reptiles and amphibians and fish have in common. Mm-hmm. And we have them too, but they're not as important in our biology as they are in theirs, which could very well be the reason why these spirochetes are cleared by the nymphal ticks sucking lizard blood. That's a speculation.
I like the idea. I mean, this is another one of those. You're always searching for ideas to do research on. And can you identify this substance? Can you somehow put it out there and let the ticks, the nymphal ticks, feed on it so that we clear them of Lyme disease? This is a great idea. I'm glad they sent us that link.
Yeah, me too.
So there's actually some literature on this. This is the western fence lizard is an important host for ticks, but is resistant to Borrelia. Its immune response to the bacterium is so powerful that it can actually clear the infection from the midgut. So actually, I would expect that there's something produced in the midgut,
Me too.
similar to this protein that I just told about ticks make that reduce Borrelia, and that is it is an enzyme that cleaves the peptidoglycan of the outer membrane of the bacteria, and so it breaks it up.
So on an interesting note, this is a side note, but nonetheless, it was one of your picks of the week once, and you helped me assemble that website on DespommierPhotoArt.com. In the gallery on animals, you will see a picture of the western fence lizard.
And I have the article for you. Danny, because you wanted to learn more. A comparative study of mammalian and reptilian alternative pathway of complement-mediated killing of the Lyme disease spirochete. So apparently it's complement.
Very interesting.
I will put the link to that in the show notes for you, Danny, so that you can read about it. And if you can't get the paper, let me know. We will get you a bootleg. We will get you a copy. All right, that'll do it for TWiP number 83.
83.
We're going to get to 100 this year because of Daniel.
I think we are.
He's given us a reboot. TWiP reboot. You can find all the episodes at microbeworld.org/TWiP. You can also find them on iTunes. You can find them at that NSF site, Science 360, I think it's called.
Nice.
You can find it on your favorite podcatcher. And they're all free. All of the episodes are there, all 83. And if you have questions and comments for us, please send them. And please try the case study.
Exactly.
To TWiP at twiv.tv.
Got nothing to lose and everything to gain.
TWiP at twiv.tv. And P stands for parasitism, which was Dickson's idea, even though he will not own up to it. Dickson Despommier, one of the three musketeers here, is at Trichinella.
I'll be Athos.
Trichinella.org. I don't know their names. Thank you, Dickson.
You're welcome.
Daniel Griffin is right here at Columbia University Medical Center. Thank you, Daniel.
Oh, it's been a pleasure.
Always fun. And I'm Vincent Racaniello. You can find me at virology.ws. The music that you hear on TWiP, I really like. It's composed and performed by Ronald Jenkins. You can find more of his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.